Thank you very much for coming to listen to a talk rather than taking a siesta time, because the heat outside is certainly more like taking a siesta now. I would like to talk about a topic where I feel that physical scientists and nanotechnology can make a huge difference. And they can make a huge difference because we are trained to think at the nanoscale. We are trained of using physical techniques, developing a lot of new tools. And new tools allow to address questions in different ways from how they have been traditionally discussed. And so the first big question is how to hang on. The art of forming and trusting a bond. So at the macroscopic scale, what does it take to mount a certain weight and support a certain weight by a bond? In a similar way, I could hold on to this piece hours. But if I would hang on this piece, I could maybe hold on for just a few minutes. So what it becomes very, very evident, the bond lifetime is not something that is defined by the biochemistry of the bond. The bond lifetime is also mechanoregulated, and typically the bond lifetime is shortened if you mechanically pull on it. So there are, when it comes to bacterial adhesion, when it comes to cell adhesion to surfaces, a huge number of questions that relate to understanding the biochemistry, but then also to asking mechanical questions. And I would like to give you a few examples of that. The first one, and maybe you or some of your relatives are suffering from time to time with urinary tract infections. What you see here is are just a scheme, E. coli attached to the urinary tract. No, I can see it. To the urinary tract. And certainly they can still attach there despite the fact that you regularly try to flush them out. So how do they do it? It's not just a question of whether they form a bond and of the biochemistry of forming a bond, but how do they hold on when you try to flush them out? In a similar way, catheters very rapidly get infected. Those are synthetic molecules that we do not even pre-coat with molecules that promote their adhesion. And what is very troublesome is, in the moment, is that the number of urinary tract infections is high, and about 80% of urinary tract infections in hospitals are due to catheters. So urinary tract infection is the most common infection that you get when being in a hospital. So since the first line of defense is our own immune system, we should have a look at how immune cells try to pick up bacteria if they firmly adhere to a surface. What you see here on the left side is a macrophage, and on the right side you see 2 E. coli bacteria. So each of these E. coli is about close to a micron long. If you look at the work that a macrophage is doing, you see the macrophage reaching out to the bacteria, grabbing bacteria, lifting them off the surface. Now it continues running. Lifting them off the surface. Also, the macrophage, or my computer is taking siesta times. And you see the macrophage is reading out. Many phagocytosis events occur. But at the end, and I hope you can see that the movie is halfway through in the moment and gets stuck, you see that finally the surface is covered with more bacteria than there have been at the beginning to start with. So the question of whether you get an infection or not is not whether you are exposed to these bacteria. It's a question of rates. how fast can the macrophages clear out an area, and how fast do the bacteria divide. And E. coli typically divides every 20 minutes. So if you are exposed to dirty water, it's not the question of exposure, it's the question how much do you drink, and can your immune system clear out the bacteria faster than they can divide. And only then do you have a chance to win against the infection. So in all these cases, it's not just making a contact with the bacteria, but it's also a question whether macrophages are able to grab the bacteria, pull it off a surface, and phagocytose it. So let me show you now a few nice other examples. First of all, many bacteria and also other microbes, they can not just exist as individual bacteria, but if they divide, they might not completely separate. And if that is happening, these bacteria can form very long filaments. And these filaments can be many microns long. So one of these filaments is here false colored green, and the individual bacteria are false colored yellow so that you can distinguish them. Now have a look what the macrophage is doing with this long filament here. And the question is, does filamentation of bacteria protect them from even being taken up by macrophages? And you will see later on this question has relevance for drug delivery as well. So you see how the macrophage reaches out, it phagocytoses the filament goes inside the cell, and then if this wouldn't stop all the time, then you would see how the filament is disappearing in the macrophage and phagocytosed. And all the bacteria that made up the filament are phagocytosed all at once. But then there are other situations. And here macrophage approaches another of these bacterial filaments. And it's working in this video for more than 40 minutes. But the macrophage is approaching the filament from the side. And it's pulling on the filament. So there are a lot of adhesive interactions. But the macrophage finally cannot phagocytose this filament. What the macrophage fails to recognize is that it just has to move a little higher up to reach the pole. And what we see in all the videos is that only if the macrophages can reach a pole of the filament, in those cases, phagocytosis can be initiated, but not in all the other cases. And even the force they apply is not sufficient to break these filaments apart into individual bacteria. So indeed, under certain conditions, filamentation can protect bacteria. So why is this important, and why should we know about that? In the design of many biomaterial surfaces, titanium dioxide, for example, a hip implant, we were thinking of roughening surfaces. And we were always thinking of roughening a surface so that we promote integration of an implant with the surrounding tissue or speeding up of a healing event. So what I would like to show you now in this video, though, is if you have any type of heterogeneities on a surface, in this case, we just made a non-adhesive stripe next to an adhesive stripe, where a pole of a filament can be hidden. That allows a bacterium to escape then an immune response or an attack by a macrophage. And you see this nicely on this microfabricated surface. Where
the laser stops them.
Ha. Okay. Okay, that shouldn't be. Then I will just. Okay, then I will just do without the laser pointer. You see in the moment on the right side, the macrophage, you see the pole of the filament extending over this chemical edge, and the macrophage is not able, despite the fact that it applies a lot of force, to reach the pole, and again, the filament can escape attack. What we can see as soon as the macrophage reaches the pole, though, is that the macrophage can very rapidly form a phagocytotic actin cup around the pole, and with that, the phagocytotic event can be initiated. What is very interesting and I think important for us in the nanotech community is that there have been studies where people ask the question whether drug delivery systems should always be spherical, or whether drug delivery systems have other pharmacokinetics if they are elongated. And so people started studying whether or not if you engineer a number of different high aspect ratio shapes, whether that promotes or slows down phagocytosis. What we see if we compare the published data with our own data is that essentially all the same rules apply, whether a macrophage is interacting with a bacterial filament or a synthetic particle that has a completely different surface composition. And the question is only whether or not it can reach the pole. And once the phagocytotic event is initiated, we even see that phagocytosis is happening with the same velocity of 8 micrometer per minute in all these cases. And that is then governed by cytoskeletal restructuring events. So looking and comparing in the moment, I think a lot of our synthetic systems with biological systems that have the same size ranges can, I think, for the future teach us many, many important lessons. So the next question is, what I've shown you so far was at the micron scale. What is happening at the nanoscale? What kind of adhesins do bacteria develop? And how and what can we learn by understanding the mechanics of these adhesins? How can we improve on some questions in medicine, but also think about maybe the making of synthetic adhesins? The first one is very tightly correlated to the question that I mentioned at the beginning. The lifetime of most non-covalent bonds is very rapidly decreased if you pull on a bond. So the strongest bond that is a non-covalent bond that many of us use in biotechnology and nanotechnology is the biotin-streptavidin bond, and we use that because the lifetime is a day or two. However, if just a single motor protein would pull on the bond with 5 piconewtons, the lifetime is reduced to 1 minute. And often we do not consider that when functionalizing surfaces with biotin-streptavidin as a coupling agent. So if you see, though, that the lifetime of bonds can be reduced to minutes only, you wonder, how can a bacterium ever even adhere to a surface? How can a big cell that immediately starts pulling with the motors on the integrants adhere to the surface? What mechanisms are in place to deal with this lifetime shortening? So it was a couple of years ago that we were working already with E. coli. And what we noticed was, against everything that we knew at that time from biophysics, we noticed that E. coli has the ability to accumulate on a surface if we flush the surface. So under low flow conditions, we essentially saw no bacterial accumulation whereas at high flow conditions, we saw considerable accumulation of bacteria on the surface. And what should you know about how this is happening? So first of all, E. coli has long hair, and at the tip of each of the hair is a little glue molecule. It's called an adhesin. And this adhesin has a binding pocket for sugars, and among the sugars, just specifically to mannoses, to monomannose. And so in these studies, we had coated the surface with monomannoses. What we then saw was that if you coat the surface with an antibody that recognizes the same adhesin, but it binds to the outside of the binding pocket, not inside, then the lifetime is really shortened as expected with time. However, this accumulation only occurs if the E. coli is seeing mannose presented at the surface. Many of our proteins are glycosylated, expose mannose. Many of our cell surfaces expose mannose. So it's a great way of E. coli to adhere to tissue and perhaps invade. So many years of work allowed us to figure out the structural mechanism that is behind this phenomena that is called catch bonds now. Here again, you see now the structure, high resolution structure of the hair that was resolved by my colleague at ETH, Rudi Glockshuber. We resolved this structure now, and in this paper, and you see that there are two domains that build the end of this long fimbral complex, and this outer domain carries the molecular binding site. What is happening is the following. How can a bond get stronger if you pull on it? In the macroscopic world, we very much know that there are bonds that can get stronger. And those are, for example, these finger traps. As soon as you pull on them, the texture tightens around your finger, you get trapped. So that's a macroscopic analog of a finger trap. At the nanoscale, similar structures exist. However, here, you see under equilibrium that these two domains are in contact to each other. We also know from the field of biochemistry that cofactors that bind far away from the binding pocket can regulate binding strength. So what we found out now is that the second domain acts as a cofactor. But in case of catch bonds, the cofactor is mechanically removed, which is different from a soluble cofactor. And once the cofactor is removed, this regulatory domain is removed, the structural perturbation propagates to the binding pocket, switches it to high binding, and the lifetime with that event is increased by several orders of magnitude. So the question is, how does it impact us? Should we care about it? What are the consequences? Again, dealing with this problem of bacterial adhesion on infections and the fact that antibiotics are not designed any longer by the pharma industry because the mutation speeds are much higher, faster than new antibodies can be developed. There's a series of new molecules in clinical trials that essentially try to prevent bacterial adhesion in the very first place. And most of them play around with these binding pockets. And if you can block the bacterial adhesins, then maybe the bacteria cannot adhere to wound sites. However, if you now add such soluble inhibitors to our flow cell experiments under intermediate shear, you still see that if we add these inhibitors, the number of bacteria on the surface is decreasing very rapidly. If we add them at the high shears where the bonds are maximally activated, there's no effect of the soluble inhibitors. It turns out in all the screening assays, people have done them under static conditions, saying fluid flow would only accelerate the detachment. What you see, though, very clearly here, though, is they're ineffective. And the soluble inhibitors can't compete with this long-lived bond that the bacteria form in the catch bond state. So you developed a soluble inhibitor, but addressing the wrong state, because you did not even know that there were two states available. This is how it's good to be motivated, stimulated by intuition, but it's also important to ask questions, how far does our intuition go if we try to predict phenomena at the nanoscale? So the FIMH was the first catch bond characterized. It turns out that integrants and selectants also form catch bonds. And in all these cases, there's always the binding domain, second domain, and close physical contact to the binding domain. This is the same for selectants and integrants. And from all the kinetic data that are known today, it also seems that in both of these cases, allosteric regulation controls and allows them to form catch bonds with their respective ligands. So let me come to a second type of bacterial adhesins. Also, that one is mechanoregulated, and that deals with S. aureus. And S. aureus, again, the theme is the rapidly growing bacterial resistance against antibiotics. The numbers are really, really stunning. About 2 million cases of hospital-associated infections occur per year, resulting in approximately 90,000 deaths. And that is the number only of 2007 in the US. The annual cost to hospitals exceeds by now already 30 billion. So it's very clear we need to make major efforts to find solutions. It's a daunting problem right now. There are a lot of pictures that you find in the literature looking just at how bacteria adhere and counting the bacteria adhering to surfaces, but not asking in detail about adhesion mechanisms. This is a map I found recently showing Europe, and and it's the probability of finding antibiotic-resistant S. aureus bacteria in human blood in several countries in Europe. And you see the probability is in some countries already higher than 50% in Portugal. In some countries it's quite low. First of all, you have huge discrepancies between European countries, but it also means the probability of each of us having several antibiotic-resistant bacteria in our blood is very high already. And the rapid increase is probably due to the fact that we eat so much meat of animals that are treated by antibiotics throughout their upbringing. And it's a major breeding system for antibiotic-resistant bacteria. So our exposure is tremendous. So how does that work in case of S. aureus? So S. aureus can adhere to tissue fibers, and particularly during wound repair and in healthy tissue, the fibers are stretched. So let me come back. If you scratch your skin and look right underneath the skin, you find a lot of connective tissue. And this connective tissue is made out of collagen and fibronectin. And in each wound site, the production of fibronectin is highly upregulated. In wound sites also, you know that fibers are cut either by a knife or by biochemical degradation by enzymes. So the first cells that come into a wound site to repair a wound, to make new tissue, those cells are called fibroblasts. And they do so by taking the fibers all around and stretching them again. So first of all, I would like to show you how fibroblasts can indeed stretch extracellular matrix fibers. You see them here, the blue fibroblasts, how they grab single fibers, how they stretch these fibers, they let go, they stretch and let go. Just once again. So these green fibers that you see here, they are made out of this protein fibronectin, shown below here. And the bacteria have long polypeptide chains that extend from the body. And these polypeptides can recognize the N-terminal domains of fibronectin. And this is just a sketch. Here you see a high-resolution structure of, in gray, fibronectin and the bacterial adhesin that recognizes not just one domain, but several of them in sequence. So in that sense, it's not just a receptor-ligand interaction that is mediated by a single binding pocket. It's more like two Lego blocks being pressed against each other. What I would like to show you now is that the bacterial adhesion peptides bind more tightly to mechanically relaxed fibronectin fibers. But stretching of the fibers can destroy these binding motifs. And the way how this is happening, we have shown that by stretch assays, but I would like to mainly show you a steered molecular dynamics simulation. That was our first simulation that indicated that you destroy a multivariant binding motif simply by stretching one molecule. And here you can see how the bacterial peptide binds to one domain, to a second domain. It forms a lot of force-bearing backbone hydrogen bonds. And then if you just pull on the gray structure on fibronectin, you see indeed how the connection to the domains can be destroyed. So we could show the same by synthesizing these bacterial adhesins that were known. The complexes were known. We could run simulations. Then we could label these peptides with one color, bind them to fibronectin, stretch fibronectin, and indeed show that the binding is reduced. What is important is fibronectin fibers are relaxed in wound sites. So with the polypeptide adhesin, a bacterium can not only recognize whether or not the protein is there, but it can distinguish between the physical state of this protein. So the experimental assays we did to demonstrate that were based on data from bioengineers in the 90s who pulled fibronectin fibers out of droplets. And we used the same technique to pull fibronectin fibers out onto silicone sheets and then stretch the silicone sheets. And then if we photo-labeled fibronectin with one color and then the bacterial adhesin with the second color, we could do ratiometric imaging of these silicone sheets. And what you can indeed see is that essentially no bacterial peptides bind to the highly stretched fibers, but many of them bind here in red to the fibers perpendicular to the strain axis. And you can quantify these strains because you can externally adjust them. And we see that at a strain of 100 to 150%, the bacterial adhesins are destroyed. So coming back to therapeutics, can we utilize this information? What is very, very interesting is that not only S. aureus targets this N-terminus, but many other bacteria do too, including this little beast that causes Lyme disease or borreliose. It also targets the same region. These bacteria have no binding sites for collagen. They only target fibronectin. But then it turns out that the same region has binding sites for fibronectin itself. So if you cleave off this region of fibronectin, fibronectin cannot even assemble into fibers any longer. And it has binding sites for heparin. Heparin is a molecule already that we use heavily under surgical conditions. Nobody ever looked into the possibility that heparin could perhaps reduce the number of bacterial infections. We try to find data and try to start collaborations with hospitals to looking into this. Because what we see is that if we repeat these stretch assays, but in the presence of heparin, you see that in the presence of heparin, we can reduce the binding to relaxed fibers, but also to stretched fibers. you further reduce the binding if you stretch the fibers. So the mechanosensitivity of the interaction is not perturbed, but heparin directly competes with the bacterial adhesion for these binding sites. So what else can we do with this information? When going back to the protein databases, we could find altogether 32 adhesins now that all recognize fibronectin. And if you start doing a sequence alignment of all these bacterial adhesins shown here, you see that there's very little sequence homology. But still, from the few data that were available, we could identify which of these amino acids will form secondary structure with fibronectin. And then you, after alignment, you see that there are always peptides that tightly bind to the domains, but then there are also regions in between, and they differ very much in their length. And they can form some of this loopiness in between. And it's the loopiness in between that determines whether or not the bacterial peptide can manage a structural mismatch that you induce by stretching fibronectin. So based on that, we see now that S. aureus is indeed the adhesin that has the highest mechanosensitivity. The adhesin that causes Lyme disease, though, has very long loops connecting the templates that bind to fibronectin. So we think that it has no mechanosensitivity in its interaction with fibronectin. And in the next step now, we can use these. We then engineered a number of peptides with short and long loops just to play around with this theme of engineering mechanosensitivity in a ligand-receptor interaction. And the loop length indeed controls the outcome. And we are developing this now as a new platform to the design of probes that can probe the mechanical strain of extracellular matrix proteins. Why are we so keen in determining some of the strains quantitatively in cell culture? This is the key question that interests us in every one of our research projects. It's how can mechanical force switch the structure-function relationship? Because the structure-function relationship so far has only been put together in a kind of one-dimensional way of thinking, that you take a structure, you take the function, you map out interactions, you put together signaling networks, and you finally hope to do quantitative biology predicting cell phenotype. But in none of these cell signal networks would you find force as a regulatory mechanism. So if force can switch the structure, it will also be able, and you've seen now examples, it will also switch the function. And then you might switch into different cell phenotype networks just by using mechanical force. And the question is, how do cells utilize this in a systematic manner? And how can we maybe, by understanding how forces can switch protein function, also understand how cells recognize material properties, surface textures, and so on? And how do they recognize, in general, their environment, the physical features of the environment? What is certainly known is, these days is, as soon as you expose a surface coated with proteins, the cells form adhesion sites. The formation of adhesion sites has been thoroughly studied by biologists. Then they rapidly reorganize their cytoskeleton, and again, a process well studied by that community. The next step is somehow, and there are many question marks to that, somehow signals from outside here lead to a change in gene expression, and finally to a change in protein expression. How this is happening is a big, big open question mark. And this comes back to some discussions here already. How do you probe for that? Because people understand these phenomena and the rearrangements of the cytoskeletal because those happened at the dimensions of optical microscopy. the nucleus is so densely packed and so much smaller than the cell that we need nanoscopic resolution techniques to look into what is happening in the nucleus. And so there are many question marks that are still open. So Dave also mentioned in the morning this paper of Dennis Disher. Stem cell differentiation is directed by substrate rigidity, and he put out some question marks. The community in the moment thinks that this is indeed true. Differentiation is directed by substrate rigidity, but the only thing that was shown in that paper is stem cell differentiation is correlated or correlates with substrate rigidity. And what these studies have done was that substrate rigidity has been adjusted by changing the polyacrylamide cross-linking density. Polyacrylamide, though, is a network. And if you change the cross-linking density, you also change the mesh size within a network. And then the second part that has been done was since cells don't like polyacrylamide at all, you covalently couple proteins to the surface of a network. So the cross-linking changed the rigidity, and therefore it was assumed rigidity directs stem cell differentiation. So there are other polymers out there, though, where the major features of the polymer are not network-like. And one of these is another substrate that we often work with, and that it's polyacrylamide, PDMS. And what I would like to show you now are data comparing tuning rigidity of polyacrylamide and tuning the rigidity of PDMS. In both cases, though, coupling the proteins to the surface in a covalent manner. Essentially, the entire community used proteins that were covalently cross-linked to polyacrylamide, and when adjusting the rigidity on PDMS, they only fusisorbed proteins to PDMS, but didn't use the same cross-linking procedures. But what is happening now if you cross-link the proteins to both surfaces? If we essentially repeat the same experiments that were published in the Disher paper on polyacrylamide, adjust the rigidity, then you see nicely the oil red, so adipogenesis here. These are fat cells. The number of these fat cells declines rapidly as you go to more and more rigid surfaces. These blue cells here are osteoblastic lines, and you see that the number of these blue cells rapidly increases with substrate rigidity. So that follows what people have thought in the community. If you repeat now with the same stem cells, with the same chemistry, this experiment, but on PDMS, you see no change. You see no change in the number of adipocytes, in the fractional number of osteoblasts. So essentially, the cells do not see the rigidity change that is happening underneath. You see that the number of adipocytes on PDMS is quite close to the number of adipocyte on rigid polyacrylamide gel. Now, in these cases, if you, the chemical cross-linking is done by light exposure, it's a photo-initiated reaction. If you just shorten the time of this reaction, fewer proteins are cross-linked, and then suddenly the PDMS looks softer. And so you can imagine if you now just fusisorb the protein, then the cells can also harvest and pull off the protein from that interface. So the cells obviously don't automatically feel the rigidity of the substrate. There's something that is happening to the protein layer in between. And the model we propose in this paper now is that if you deal and start with a network and then covalently cross-link proteins to this network. In case of a large pore or mesh size, the protein can be deflected a little more. It's not just a question of ligands that are exposed, but the protein itself can be deflected more. Whereas the protein here is firmly anchored if the mesh sizes are much smaller than the length scale of the proteins or the protein fibers. And so it's the way of how the extracellular matrix is coupled to the substrate. It's the physical properties of this protein layer that the cells feel. It's not the rigidity. But in tissue engineering now, if you try to optimize a material to a certain cell function, we should not assume that it's just rigidity that we need to optimize. We need to think about what is a correlation and what is it that directs cell function. So coming back to this scheme here, what we are extremely interested in how do the cells respond to this protein layer. And once they adhere to this protein layer, they also secrete and assemble a lot of extracellular matrix fibers themselves. And very rapidly, they don't feel the proteins any longer. They feel their own matrix. And in a moment, we try to find out what is happening with the matrix that they make themselves, and how soon do they sense the matrix that they make themselves. Because they can stretch proteins, but then they can also stretch the matrix fibers, and I've shown you already examples how much they can stretch the matrix fibers. Another illustration how the stretching of a protein can change its function is illustrated here by using antibodies against fibronectin. So very often antibodies just have one binding epitope. But in many cases, unless you really know your antibody well, the antibody binds to more than one epitopes. So for example, an antibody can bind in between these two domains. And here I would like to show you that if you do an antibody binding study and see dark, it doesn't mean the protein is not there. So we prepared the same stretch assay. Our fibronectin fibers were stretched this time in a vertical direction. You see the bluish green color. As you, no, you haven't seen it before because I showed that slide only on Monday in the biophysics part of the meeting. So the color here in the stretch assay comes about by us introducing small amount of Fred-labeled fibronectin. And high Fred corresponds to the red color. So you see here in red the relaxed fibronectin fibers, and the highly stretched fibronectin fibers have this bluish green color. And if you then in a third channel label the antibody with a third color, you see that the antibody binds only to the relaxed fibers, but not to the stretched fibers. So even some antibodies can sense and are mechanosensitive. So if you deal and try to stain a cell sample with antibodies and you see no color, it doesn't mean the protein isn't there, particularly if the protein is part of a force-bearing protein network. It can simply be that mechanical force stretched that protein and destroyed the binding epitope. And what proteins do you and should you keep in mind with that? I just show you here the junctions of integrins that cells form to connect the cytoskeleton to the extracellular matrix out here. You see integrins, the alpha in blue and the beta subunits that couple to adopter molecules, and the adopter molecules then directly link to the actin-myosin system. And so force applied by myosins can pull on all the proteins that are part of the force-bearing network. You've seen already how the myosins pulling here can stretch fibronectin fibers. But also every protein in between can be physically stretched by force. So what I said with respect to antibodies might apply to many other proteins as well that are part of these force-bearing networks. So how do we go now from studying cell behavior in 2D to making this transition to what can we learn for tissue engineering? So what is very important for us to learn how we can engineer complex 3D microenvironments, but microenvironments that are not just synthetic polymers, but microenvironments that mimic some basic features of tissues. And I would like to show you, we have two approaches ongoing in the lab right now. The one is cell-derived 3D extracellular matrices, and another way of making microtissues. So the best healing features so far in tissue engineering come from decellularized tissues used as tendons and so on. But understanding and imaging cells in decellularized tissues, we tried that a number of times. It failed. There was so much autofluorescence in these samples that we couldn't do at high resolution any studies. So Ken Yamada, a couple of years ago, published a paper where they described how they grow fibroblasts on the surface, culture them for an extended time period, then they make matrix, and later on you can wash out the cells from this matrix and reseed this matrix by cells of interest. So in our case in the lab in the moment, we reseed them with fibroblasts, stem cells, cancer cells, immune cells, and simply ask how these 3D environments regulate their function. But the very nice part now is you can add in trace amounts in this culture medium already photo-labeled fibronectin. And what you see here is those are not antibody stains, but here we, for example, inserted into the medium green labeled fibronectin, and you initially saw the video of that sample. And then as we reseeded the sample with cells, we added now red labeled fibronectin to the medium. By that we can see how does the cell interact with the old matrix, with the old scaffold, and how does it put new fibers into this old scaffold. And you can see it's not like many onion shells of red and green fibers, but the new fibers are really nicely interlaced with the old scaffold. And furthermore, we can now also add then thread probes into the old and into the new and ask how it all works together and what the local strains are that cells are exposed to. So that's one model system, and we hope to get a lot of data out of those in the near future. A second system was essentially introduced by Chris Chen and his student Wesley Legend. And the approach that Wesley took was he engineered micro pillars in a small microfabricated chamber. Then he filled this chamber with a collagen, 3D collagen gel. And in between this collagen were fibroblasts. Initially, they were completely isotropically distributed. This entire chamber was filled with collagen gel. But as the cells started to contracting, they contracted the entire collagen gel. And after 22 hours, you see that a microtissue had formed. Here's the collagen gel. The tissue lifted off the substrate, and finally you could even measure the strain of the tissue by the deflection of these posts. So Wes then came to our lab to finish his last year of PhD with us in Zurich, and we asked essentially questions: how do they form new tissue surrounding this collagen gel? So collagen gels are often used in cosmetic surgery to fill places. So if there's collagen gel, how do cells deal with these collagen gels? So what you see after several days is, for example, a microtissue where we have a DAPI coloring for the nuclei of the cells, then in green color fibronectin, and in this pinkish color collagen. So in detail, what is happening during this remodeling process? So first, the cells sit really in this collagen matrix, And in the moment we zoom from the top with the confocal microscope through the microtissue to the bottom of it. And you see already in these scans the moments when where the cells show up is greatly different. If you look at these videos a little longer, what you will see is that after 72 hours, the cells are not dispersed in the collagen any longer. Essentially, all the cells have left the core of this contracted collagen gel. They are sitting on top of the collagen gel. And on top of this collagen gel, they now start making a new fibronectin matrix. And then we even see cases where the new fibronectin matrix is then slowly complemented by a new collagen matrix made on top. So it's not a homogeneous medium. It's highly heterogeneous. What is also interesting, since we are so interested in stretching of fibronectin, were then further studies where we asked what kind of fibronectin population, how much is fibronectin now stretched throughout this microtissue. And as soon as you expose a collagen gel to fibronectin solution, a lot of these proteins in a globular state adsorb to the collagen fibers. And here now we ask, how many of the fibronectins co-localize with collagen? That is the red curve. And how many of them do not co-localize with collagen? So all the blue fibronectin molecules are within fibers that the cells made in response to experiencing the collagen environment. And you will rapidly see as we scan now through this microtissue that we have two populations of fibronectin. So both of these curves are fibronectin curves, but you see that essentially the fraction of just adsorbed globular fibronectin molecules to the collagen, it persists throughout the time. The cells are not able to remodel that fibronectin any longer. They only remodel and make new fibronectin at the periphery of this microgel. So the physical state of fibronectin inside the microgel and on the outer surface is greatly different. And also the conformation of fibronectin does not correlate with the major strain axis throughout this tissue because the major force-bearing elements go through the central part of this microtissue. So since many of you are engineers, we addressed a lot of biological questions. There are some lessons for you to learn as well by seeing how mechanical forces can be utilized to regulate bond strength. And I think the most important lesson for all of us to be learned out of studying the mechanics of proteins and protein-ligand interactions is the fact that mechanical stability is not equal thermodynamic stability of a bond of a protein. And the nicest way to illustrate that is, how would you open a zipper? You would open it step by step by step. And if you do so, you don't need a lot of force. If you try to open a jacket this way, you need considerable amount of force. Whether you have such a geometry of molecular interactions or this shear geometry, thermodynamically, you only form in both cases 4 or whatever number of bonds. So in what regulates mechanical stability is really the geometry by which you open an interaction. And this has major, major implications in the way of how we should design mechanical systems, microfluidic systems that are functionalized. And to give you a few examples here, using DNA to link molecules to surfaces, and there's a very nice study done, a systematic study done from Hermann Gaub's lab, showing that you indeed, if you functionalize a surface with oligonucleotides, here the geometry is in a zipper geometry. This part can be removed very easily. Here the blue part is in a shear geometry where you have to break all bonds at once if you want to separate the upper and lower parts from each other. We can use the same for to perhaps one day realize some nanorobotics underwater. What we engineered here is a map of Switzerland. And Switzerland is not much more bigger than Israel, but you have, I think, 4 times the number of inhabitants. Is it 20 million against, yeah? 7. Then we even have the same number of people in the 2 countries. But what you see here is the map of Switzerland, microfabricated. And Switzerland has perfected the ability to collect money outside of Switzerland and transport it effectively to Switzerland. And we've tried the same to see whether we can engineer loading stations for gold at the border of Switzerland, and then use motility assays driven by motor proteins to transport the gold to not Swiss banks, but to ETH, because we sometimes also need a little bit of money. And the gold were not gold bars, but just gold nanoparticles. And what you see now, this was microfabricated. ETH was printed on the surface. Here we played around with zipper geometries on the border and these shear geometries at ETH. And now I would like to show you one motility assay where the microtubules, here the little structures are motors, the kinesins. Then you see microtubules, and you heard a lot of work about microtubules earlier today. And you now, the microtubules are allowed to move randomly, but as they come in contact with gold bead nanoparticles, they can indeed transport them to ETH and then deliver them at ETH based on this much more sticky shear geometry. Here, these forces that are necessary for such transport, these numbers come from Hermann Gaub's AFM studies. And to show you the associated video, here you see a microtubule coming in and delivering a gold 40 nanometer particle to ETH. So the probabilities here were even so small because a microtubule has 30 nanometers in diameter. You need to optically visualize the trajectories, so we had needed a very sparse density of gold particles on the border, and then yet to be able to focus with highest resolution and see events, and but you see events where the microtubule can indeed pick up cargo and deliver it where it's needed. So with that, I would like to very briefly, we had a nice discussion at lunchtime about nanoscopic optical tools, and I think several of you are working on that, and several of you want to apply them. And there are so many nanoscopic tools out there in the moment. How do you pick the right tool for your question? And we developed also one tool recently in the lab to be maybe on a longer term able to look into the nucleus with higher precision, and to start visualizing DNA with nanoscopic resolution. We took dyes rather than engineering dyes that you attach to a sample that then blink or you photoactivate them. You can also use dyes that diffuse randomly in solution, and they only get bright in the moment where they bind a sample. And either the lifetime in the bound state is limited by them dissociating again or by them getting photobleached. But also through this mechanism, a binding based mechanism, you can reduce the number of fluorophores considerably and just get stochastic information about a structure without pre-labeling the sample. And here you see now an E. coli bacterium, and at higher resolution, you can start seeing some of its DNA structure. So on single strands, we could come down to seeing structures with 10 nanometers. So with that, I would like to come to a closure. outlook, insights into how mechanotransduction processes work will hopefully inspire new approaches in bioengineering, as well as to diagnose and treat diseases. And it's important for us to look in great detail how some of these beautiful natural nanomachines work and what lessons we can learn for technological applications. A lot of this work is done in many international collaborations, so we are also part of Nanomedicine Center, and I think you recognize many characters here, including Benny is in this center grant, and then we have colleagues all over the world from engineering to biology, and the people that did the real hard work that I all mentioned throughout my talk with names. These are the faces, and I am very gratefully to them. Thank you very much.
Thank you very much for the contribution. Open for questions. Yes.
Not sure it's related, but cranberries are believed to prevent E. coli from adhering to the body to prevent UTI. Are you familiar with the mechanism? Is that related to what you mentioned about the lifetime of the bonding?
You said cranberry juice?
Cranberries.
Yes.
It's not juice.
Yeah. It's certainly something that is a general recipe. We never put cranberry juice so far in our flow chamber assays. I started reading a little whether there's information about the mechanism. There seems very little. Certainly it contains a lot of different sugars, but this is, I can't say more.
It's not adhesion?
I don't know. Yeah.
Did you try manos in your flow chambers?
So all the adhesion happens to manos and also to different type of manoses. So we worked intensively with the monomanos, with the trimanos, and then some manoses engineered by Peter Seeberger. And as long as a single manose ring can slip into FIM-H, into the binding pocket, the catch bond can be activated. The role of additional manose rings can be to adhere from the outside to the adhesion, and by that
if it's
stabilize.
Just in the surrounding solution, it will not prevent?
The soluble inhibitors are manose analogs, and you've seen those results. Yes.
And which dye did you use for the DNA binding assay?
We tried several dyes, and including yo-yo.
Sorry. So, if the sort of standard screening technique for inhibitors it doesn't work properly, right? So, what do you think would be a good strategy to sort of overcome catch bonds? Right, to come up with inhibitors for catch bonds?
This is certainly a next question, and I think we are far from being there. We can, in the moment, focus on first showing a mechanism, because without a mechanism, it's very hard to really think how to overcome that. It's also important to show why a number of clinical trials did not work, and so we hope with people in industry to think about how to now pursue alternatives. But we are not there.
We found some correlation between, in clinical samples, between patients that were paclitaxel, a tubulin inhibitor, resistant, and alpha-V beta-3 integrin expression. Have you ever had any experience with cells, with cancer cells that are resistant in your systems to see their adhesive properties? Do they change? I wonder if this correlation is meaning.
We have not yet looked into cancer cells because all the ones we got were so poorly characterized. We started looking more about how the cytokines, they produce impact fibroblast function. And there we see major regulatory roles. But that's work that is ongoing right now in the lab. So we got some fibroblasts from stage 4 cancer patients and compare them with normal fibroblasts to get first a little more information what might control the changes in the stroma, which is an extracellular matrix change that is happening there.
We know that endothelial cells that are part of the stroma do express a lot of integrins, overexpressed integrins, compared to normal endothelium, so it could be the same mechanism.
That's right. I think there are so many different mechanisms all playing together, and finding out which of them controls finally metastasis formation, I think is still a path to go. It's not just rigidity upregulates metastasis. I think it's not that simple. Yeah.
Okay, I think we should conclude. Let's thank Professor Vogel again. On behalf of the Nano Center, I have a small present for you. Thank you very much. Okay, let's thank again Professor Vogel.